This week's lectures are on sponges, or the phylum Porifera, and there you see the relevant pages in your Benton and Harper textbook, as well as the supplemental reading I gave you on an interesting glass sponge reef, a living one, off the coast of Washington today. So take a look at that. First, we need to talk about this phylum Archaeocyatha. It's a unique phylum because it's extinct. It's one of the only ones. Some people would say it's the only one, but it's one of a couple that is no longer with us. It lived only in the early to mid-Cambrian, and for a long time we called them sponges, we called them algae, called them all kinds of things. If you look at that picture there, you see they have a unique sort of double cone shape. It's like a snow cone cup tucked inside of another snow cone cup with various walls or septi connecting them. And they built a very sturdy calcite skeleton. They are divided into two groups, the class Regulares and the class Irregulares. And the Regulares are on top here. And you see the two circular walls, the snow cones, and then the straight line septi. They look like spokes of a bicycle wheel going out from them. They're very regular, hence the name Regulares. The class Irregulares has the same sort of general shape. There's an outer wall, there's an inner wall or an inner area where the growth stops, but then you see there's just a mat of irregular wall, sort of spongy type material between the two. And those are the two groups, and both of them lived the entire time from the early to the mid-Cambrian. Their paleoecology is pretty unique. We think that they were probably passive suspension feeders. They just grew up into the water column. And just given their nature, if water carrying food is passing over them, it's going to billow around, go into the cup, be filtered or grabbed somehow by the eater, eating cells in there, and then spewed out the top. They lived exclusively in the marine environment. There's no evidence that were ever found from non-marine environments. There's nothing for like that in the Cambrian. And within very shallow depths. Possibly they had algal symbiosis, and we'll see that's one of the reasons why we see that they built reefs. I like these two pictures here because you have both a cartoon of the simple structure of it and then a face of a rock, how we normally see these things. We rarely see them as the whole skeleton you see here. Instead, they're embedded in rock, and we see broken, irregular shapes of these things. In your book, there's box 12.3. It's a page and a half, or probably one page total, on 319, 320, which talks about a very interesting little experiment that a colleague and friend of mine years ago, Mike Savarese, did. He built little models, and all he varied in these things was whether they had septi between the two walls or whether they had pores on the outside. And it turns out that having no pores but with septi was the most efficient feeding type, meaning water got in and then it wasn't sort of lost through the outer walls, it spewed out the top. So presumably if water billows in, it's going to get captured, the food particles in the water are going to get captured before it's shot out of the top here. They are famous as being the very first reef builders in the Phanerozoic because it's clear from a lot of them that they had a lot of asexual reproduction. There's a lot of little buds sort of coming off of mom or dad, and they built big reefs. So they're the first big reef builders. They went extinct though, so if they're so great and they did such a unique thing early on, first in there, why are they gone? And the thought is that they were outcompeted by true sponges, or porifera, which we'll show in just a second here. But first, I like this diagram. This is a famous diagram showing reefs through time. So the y-axis is geologic history. Again, we live at the very top line, and there's the geologic periods going back. The little visual graphic in the center represents different bioherms or biological buildups, herms, and the wider the image, the bigger the reef buildups. And as you can see here, we have no evidence of reefs in the Precambrian. In the Cambrian, we start getting these things, and then the best part, over here on the extreme right, you have the major skeletal elements. You have the things that built reefs throughout time. We are used to saying that reefs, coral reefs, we equate the two. Reefs are corals, corals are reefs, but that's just been for the past several decades or several tens of millions of years. If you go back in geologic time, reefs have been built by all kinds of things: clams, sponges, bryozoans, corals, you name it, you name it. And the very first were these archaeocyathids here. Archaeocyathids are a good way to show the various reef structures or reef jobs. We can say that reef animals will either bind, baffle, or frame to build up that reef. Binding is literally gluing or cementing together, and you see here two sort of types of binding, with long branchy organisms or low encrusting organisms. Baffling is where even organisms grow even taller, and what happens is their bodies will baffle or stop the flow of water and sediment, which billows around and then piles up around it. And so they slowly, by their growth and the successive piling up of sediment, build a reefy structure. And then finally, the reef job or reef rock type that is most common today is framing. Framing is, if you frame a house, you're literally setting up the foundation and the structure to build the thing. And that's what this is. This is building big, massive skeletons that form the framework for the reef. This is what corals do today. And archaeocyathids did all of these things in different ways throughout this early Cambrian period. If we look at this chart here of archaeocyathid evolution and extinction, again, time is the vertical axis. All of these different columns are spindle diagrams of different groups, and again, the width of the column is a representative of the overall diversity of the column. Today, you see we have three different major types of sponges around. We'll talk about a fourth when we talk about sponges in particular. But then look way down here in the Cambrian. There's the archaeocyathids, and they were really wide. They were everywhere, and then they pinch off. And again, this is a good example of the wedge where one organism is diminishing, going away, and finally blinks out as another group of organisms expands, gets more expansive, more abundant, and the thought is that it is at the expense of this one. So let's talk about these sponges, the poriferans that outcompeted these archaeocyathids. There you see a picture and a cartoon. They're very, very simple organisms, and I hope you watch the little Shape of Life video from the link I gave you in the Canvas module to talk about these things. They have very simple body walls, 3 layers of cells, and they've been around, I say here, Cambrian to recent, but there's now some evidence that they were in the Precambrian as well. Paleoecology, pretty simple. They are benthic, epifaunal, immobile suspension feeders. They're found at all latitudes, all water depths, all salinities of water, from fresh to marine to very salty water. So very, very interesting group. I like this picture here because look at the blue pattern around this generic sponge. Water is going in, it's being sucked into the sides of the sponge and being spewed out of the top. This is the basic feeding mode for all of these poriferans. Water carrying food goes in. There are cells, these choanoflagellates, that grab the food, and then the water that is spewed out of the top has no food or less food. The basic skeletal plan has 3 different grades, from the simplest, which is this ascon to psychon to leucon. And really all you're seeing here is that the inside of the sponge gets more and more complex, more and more highly invaginated to add more surface area for more of those choanoflagellate cells to grab food particles. The cell walls, or the skeletal structure, is built two ways. Either there are free spicules, we'll show those in the next slide, or spicules glued or fused together to build actual walls. And then we're going to talk about their multiple mineralogies in just a second. The spicules themselves can be described, and again, there's no scale here, but these are generally microscopic to several millimeters in size, and you see they have all different shapes of, sort of spears and hooks and anchor looks, and they look like those jacks from those old games, all these things. Of course, being paleontologists, we've got different names for them all. I just hope that you recognize spicules. This is one way to build a skeleton, the simplest way. So have these embedded in soft tissue. If you then have so many of them, you can fuse them together to make actual cell walls or skeletal walls. Several classes. First, the hexactinellids, and these are the glass sponges. They are those sponges that built their skeletons out of silica, so SiO2. Very interesting group, and sometimes they're called the glass sponges because glass is also made of silica. And here's where, again, I recommend that you read that Dibus 2008 article that I put up for you. Interestingly enough, in the Paleozoic, or PZ, they were found at all water depths, all across the continental shelves, into shallowest water, into the deep. After the Paleozoic and the end Permian mass extinction, they tend to be found only in deep, deep water of the Mesozoic and Cenozoic. The next group are the calcareous sponges, the class Calcarea. They build their skeletons out of calcium carbonate, calcite walls. And they are famous for building giant reefs like these ones here in West Texas. Then we have the class Demospongia, and they built their skeletons out of an organic molecule, spongin. It's very similar to the molecule that we build our hair and our fingernails out of, and they're the dominant group today. So if most people think about an ocean sponge, they think of demosponges like this, and they're both marine and freshwater sponges. But then finally there is a group called the Sclerospongia. And they have a geologic range from the Ordovician to recent. We thought that they were completely extinct in the Mesozoic until people started finding them in, when I say cryptic habitats, basically I mean underwater caves in the Bahamas where they still exist. They built giant reefs in the Devonian. So if you go back to that reef diagram, reefs through time diagram, you'll see that one of the largest reef development time periods was in the Devonian, and the big reef builders were these sclerosponges. Now, the three previous classes of sponges were building their skeletons out of specific mineralogies. These things, these sclerosponges, or their stromatoporoid is a subclass within them. This is the fossil group, the subclass. They have a two-part skeleton. The basal part is calcite, and these are made of layer after layer of all these laminae. So these are layers, laminae, and you can see the names here. An individual one's a lamina, and then you have the various galleries and latilamina, etc., etc. In the upper surface where it's growing, the soft tissue of the sponge will also produce isolated spicules of silica. So that's extremely rare. Usually in the oceans, or always in the oceans, things either build their skeletons out of one or the other. This is the only group that builds them out of both. Interesting structures, and you'll always recognize them by this very dense laminar structure, but then sometimes if you have an upper surface, you'll have these astrorhizae and mammillons, same root as mammary glands, breasts. They look like little breasts on these things. And then astrorhizae are the little sort of drainage canals on the top of these things. Again, just some more pictures of these things. You have horizontal laminae, and then you'll also see vertical lines that we call pillars. Stromatoporoids are a good time for us to talk about plastic colony growth. It's going to work for these sponges, but it's going to work for other sponge classes, for corals, bryozoans, basically a lot of these colonial blobs that lived in the ocean throughout geologic time. So due to environmental variability, the same species can have many different shapes. It can be very flat or laminar, like this upper picture here. Notice this is practically horizontal, looks like a stack of papers. Or you can have them build very bulbous domes. They grow upward, like the lower picture here. And so this is how we describe the colony shapes for these things. We will measure the vault, which is basically the height of the colony. And these things can be big, so sometimes you have to measure it in meters. The ones that we're going to have in labs, we're going to measure in inches or many centimeters. And then the base, which is the bottom, which measures the width of the organism. And so then we divide the vault by the base, V over B, like it says here. And then you have these different categories. If you have a vault over base that is much less than 1, so less than 0.1, that's essentially growing very flat. The width, the base, is much greater than the vault, the height, and so we call that laminar. And then as it starts growing up, it becomes low domes, high domes, and then extended domes. And if you have anything that has a V over B that is greater than 1, we call it an extended dome. Now what's important about that is that this VB, or vault base relation, has to EOD, to environments. So high water energies grow laminar sponges, laminar corals, laminar skeletons, and low domes. And then low water, these things can grow up. And so again, if you look at this picture, we've got laminar, low domes, and then they become higher domes and extended domes. And you go in a spectrum from high energy all the way to low energy. We'll talk about these other features here, the sort of edge of the organism, later, but this for now is very useful. And that ends our discussion of sponges.